Effects of Natural and Artificial Light on Microcosms by Keith B. Thomas for General Ecology, Microcosm Project, October 25, 2020. Microcosm Overview. Experimental microcosms used in ecological studies address climate change, biodiversity, and trophic dynamics. For example, microcosms designed to study polychlorinated biphenyls, PCBs, transference in anaerobic shallow sediment supported quantitative characterization of the diffuse transport of PCBs in deposits within months. As demonstrated above, microcosms test hypotheses in a controlled setting. During this microcosm experiment, the main objective was the observation of trophic dynamics within a partially closed ecosystem. The experiment, Microcosm I, and the control, Microcosm II, considered in this experiment, are partially closed due to their energy exchange in the form of light inputs and heat loss. Hypothesis. In 6 weeks, if I regulate temperature in conjunction to 12 hours of artificial light, all abiotic cycles within Microcosm I will increase plant growth and its invertebrate population. Natural sunlight exposure for Microcosm II will sustain life but would not increase plant growth or its invertebrate population. Methods and materials. Several factors were considered when building the microcosm, a few of which are location of samples from an area with both autotrophs and heterotrophs, amounts of vegetation and invertebrates, container size, construction, and material. This is the neighborhood pun in which I retrieved the samples. The location of the samples were taken at MGRS 17 Sierra Papa Victor 84445 01301, a small pond used for flood prevention within a small subdivision. The pond has a pH balance of 7, which is ideal for fish and invertebrates. pH levels below 5 reduces reproduction and can result in death. September 13, 2020. pH balance reading of 6.5 through 7 was taken by a micro-essential laboratory measuring tool for pH balance. The measuring of vegetation was in length and not grams. There was no measuring of invertebrates before their placement within both microcosms. However, both microcosms received the same approximate number of visible invertebrates. Figure 1. Microcosm I and II descriptions and input. Plant length, water volume, container, sediment, indirect sunlight versus artificial light, container height, sediment height from the bottom, estimated turbidity, and room temperature. The containers of choice were glass. The choice was made after researching the best containers used to mitigate the release of energy, also known as saturated vapor pressure of the liquid. The use of glass containers prevented fluctuation in container size and integrity as well. Building both microcosms involved 2 days of preparation, from the 13th through 14th of September 2020. This was done in order to allow the sediment collected to settle within the water. The settling of sand transformed the estimated turbidity of both microcosms from 200 FNU to 25 to 50 FNU. Completion of all measurements of sand, vegetation, and pond water ensured microcosm to microcosm continuity. Measuring of invertebrates or the counting of each invertebrate was not feasible. However, each microcosm contained sea shrimp, Astracoda, and nematodes. The experiment ran for 6 weeks consecutively. External temperature of the microcosms was 22.2 degrees Celsius, excluding energy exchanges between artificial light and indirect sunlight. Microcosm I external input involved artificial light, 12-hour timer, 60-watt LEDs placed 14 inches away from the glass container. Microcosm II external input utilized indirect ultraviolet light, which was dependent on natural environmental conditions, as viewed in Table 1, from placement on the windowsill. Table 1, Microcosm II, weather conditions and area code 28304. On the left, Microcosm I, week 0 through week 1. On the right, Microcosm II, week 0 through week 1. Microcosm I, week 2. Invertebrates consisted of sea shrimp, planarians, copepods, nematodes, and tubiflex. Microcosm II, week 2, invertebrates consisted of only sea shrimp. Microcosm I, week 3, there were no visible invertebrates. However, there was substantial algae and plant growth. Microcosm II, week 3, no visible invertebrates and no substantial algae or plant growth. Microcosm I, week 4, no visible invertebrates, substantial algae and plant growth. Microcosm II, week 4, no visible invertebrates, however, there was minor algae and plant growth. Microcosm I, week 5, no visible invertebrates, however, areas in direct pathway of artificial light had substantial algae growth. Algae began to cake against the glass container. Microcosm II, week 5, no visible invertebrates, however, there was moderate algae or plant growth. Microcosm I, week 6, no visible invertebrates, areas in direct pathway of artificial light had substantial algae growth, and algae continued to cake against the glass container. Microcosm II, week 6, no visible invertebrates, however, there was still moderate algae or plant growth. Microcosm I, at the end of week 6, no change. Microcosm II, at the end of week 6, no change. Major findings. pH levels of Microcosm I were 5.5 to 6. These levels are not detrimental to fish, but could if they fall below 5. pH levels of Microcosm II were 7 through 7.5. If the pH level increased to over 9, release of ammonia can result. The hypothesis of this microcosm experiment was false. In 6 weeks, with temperature regulated in conjunction to 12 hours of artificial light, all biotic cycles within Microcosm I, with the exception of plant growth, decreased or ceased to exist. Living invertebrate populations ceased to exist from both microcosms after October 8, 2020. Natural sunlight exposure for Microcosm II did not sustain invertebrate life, but increased plant growth. Planarian invertebrates were only visible in Microcosm I. However, all visible living planarian exited the pond water and crawled upon the glass not submerged. All planarians ceased to move and expired. The most reasonable purpose for the migration of planarians and the loss of invertebrate life was the absence of aerated water. The type of microcosm constructed during this experiment involved photosynthesis exceeding respiration, especially within Microcosm I. Hypothetically, rooted aquatic biomass will increase visibility, or phytoplankton will bloom and water will turn green. A balanced system of P equal R would have accurately represented the biosphere. There are balanced microcosms where respiration equals photosynthesis. However, these microcosms were sealed and did not have aeration. 3 core concepts evident within this experiment. Number 1, productivity, i.e., generation of organic biological matter through harnessing solar radiation, the flow of energy from the sun and artificial light through autotrophs, through heterotrophs, and through decomposers. Number 2, plant adaptation to their environment. Both microcosms' vegetation adapted to their changing environment. There were visible dynamic changes between the biotic and abiotic components of the small ecological system. The experiment proved how quickly plants adapt to the increase or decrease in light, soil nutrition, and temperature. Number 3, populations cannot grow forever and are limited by environmental resources. This was observed within both microcosms after October 8, 2020. Controlled environmental resources were food availability, ecological conditions degradation of the invertebrate's original niche, and sufficient space for creature development. Experimenting with microcosms is relevant in general ecology. Controlling the nutrients placed within microcosms reflect various interactions and aspects of various organisms and their metabolism. For example, placing specific abiotic factors within a microcosm under experimental control can simulate climate change and human disturbances in the form of man-made chemical introduction. Retesting of this hypothesis should include or be altered by the following two ways. Number one, aeration of two semi-closed microcosms to annotate the effect oxygenated water has on biotic reproduction and growth. One microcosm would have artificial light inputs and the second would have natural light inputs. Number two, utilizing three microcosms without aeration of water, one with artificial light, one with natural light, and one placed in the area of absolute light. Testing of the third microcosm would demonstrate the effect internal energy inputs, if any, has on the biotic systems at MGRS 17 Sierra Papa Victor 84 445 0130.